Fighting Blindness Canada's Viewpoint is a virtual education series that brings you the latest in vision research presented by health experts from across Canada. The webinar you're about to watch is a recording. To learn more about the research we fund and upcoming webinars and events, please visit our website at fightingblindness.ca. This Viewpoint webinar is proudly presented by Bayer and supported by Allergan, Biogen, Janssen, and Novartis. We'd also like to thank Accessible Media Inc., our national accessibility partner. Everyone here should download the AMI app, which is a fabulous resource. And you can watch excellent programming, including previous Fighting Blindness Canada events. Thank you for watching. We hope you enjoy this webinar and share your thoughts with us in the comment section below.
Hi, Dr. Kian. How are you doing?
Hi, I'm doing well. Thanks.
Thanks for joining us.
My pleasure.
So as Morgan said, we had so many questions. We have so much interest from the community. So we're going to start off with a few questions just to get people a little bit more information about what gene therapy is, but we'll try to keep sort of the Fighting Blindness Canada questions on the short side so that we can really answer as many community questions as possible. So just to start us off, would you tell us a little bit about yourself and the work you're doing and how you got into this work?
Perfect. Absolutely. I think that's a great starting point. And thanks for organizing this webinar. It's my first time participating as a speaker, and I've wanted to do it for so long, as you know, and it's really a pleasure to be a part of the group today and to answer questions live. As you were mentioning, so I'm Cynthia Kian. I'm based at the University of Montreal. What I bring actually, and I'd like to share today at the webinar, is my dual interest or my expertise, which is in both retina, both the medical and surgical practice of retina, as well as inherited retinal diseases. So those are sort of the combined interests that make it that I participate in the enrollment of research and upcoming clinical trials, but ultimately I'm also the retinal surgeon who performs the surgery. So I think if people have particular questions pertaining to that aspect, the upcoming surgical aspects, that's something that I perhaps have a firsthand perspective on. Obviously, I'm part of a much larger group of inherited retinal specialists across the country that work with FBC very closely. And so the current projects that I'm most involved with include, first of all, will be at the University of Montreal, one of two sites that are administering the gene therapy for RPE65 gene mutations, the Luxturna treatment. That's one of the major projects we have on board. And then also working with my colleagues across the country to look into, of course, the registry and also other up and coming gene therapy trials that are most likely coming this year or in the next quarter shortly.
Well, there's so much to talk about just in that little opening statement. So both the first gene therapy and the new clinical trials. So just to give everybody a grounder, can you give us a brief description or explanation of what is gene therapy? So when we talk about gene therapy, what are we talking about?
So the gene therapy, especially what we've kind of been hearing through the newsletters, pertaining to, for example, RPE65 is, as we know, there are many inherited conditions, in this case, retinal diseases, that are due to mutations in genes, specific genes that code for areas within the eye, and here specifically within the photoreceptors or the retinal pigment epithelium that lead to decrease in vision since birth or continued progression or deterioration of vision. And the idea of gene therapy is basically, especially in the model we have here, is we are trying to package the correct gene product into a small vector or carrier. In this case, it's a viral vector in most cases, which is then introduced into the appropriate anatomical location in the eye, where the functional copies of the gene product or the proteins can then once again be produced and then lead to improved or hopefully maintenance of vision, which without it, with the mutation, will continue to regress or to deteriorate due to the lack of a proper functioning copy of the gene.
So a couple of things just to sort of unpackage what you're saying about the gene therapy. So you mentioned a virus. So I know we're talking about different types of viruses here, but what sort of virus is it? And I'm assuming it's obviously a safe virus, so they wouldn't be using it. But if you could talk a little bit about how that works.
Yeah, sure. So the classic, the gene therapy that's been approved by Health Canada, for example, for RP65, this is using a viral vector, which is an adeno-associated virus. As people know, when we hear virus, it's it's quite alarming sometimes. And in a way, I think it shows that science is trying to make use of the natural properties of what a virus is. In this sense, in this case, these are viral vectors that have been tested extensively, to bring the first product from basic research onto approval by Health Canada, and now as an approved treatment in Canada. This was a process that took over 20 years. So there are extensive tests and rigorous trials that tested for the safety of these vectors. And what it does is that you're able to, as we were saying, package the useful genetic information that you want to introduce into the system. And then you make use of the natural ability of the viral vector to integrate into the host or the target site where you want it to act, and then use that ability to be able to introduce the correct copy of the gene. So in a sense, there's no viral information that's of natural source that's being introduced inadvertently. This is really a very targeted approach where you know what product you're introducing, exactly where you're introducing it. And in this case, for example, this is for RP65, this is a type 2 adeno-associated virus. And we know that there's different types of adenoviruses, and type 2, type 5, type 8, for example, these are known to have a good attraction or targeting towards the structures of the eye or the retinal pigment epithelium and photoreceptors.
Okay, so these viruses are being picked because they very specially bind to the retinal cells. They won't necessarily bind to other cells as well.
Right.
And then they can put the DNA into those cells. Okay. So talking about putting the DNA into the cells, could you talk a little bit about the different types of gene therapies? Because I know there is, for instance, the Luxturna gene therapy, which is a gene replacement therapy, which is putting in a full functional copy. But then we also hear about other types of gene therapies, which are using things such as antisense, oligonucleotides, or other things like that. So can you maybe talk about some of the classes of gene therapies out there right now?
Right, sure. So as we're saying, you can broadly classify these gene therapies as those with viral vectors, those without viral vectors. So you can have vectors that are non-virus based as well. In terms of viral vectors, we talked about adenovirus, which is very commonly used for ocular gene therapy, but there are also other classes of viruses such as lentivirus. So, you mentioned the gene products. So for small gene products, which is the case for RP65, the adenovirus has the carrying capacity of incorporating that into the viral package. But for other mutations, which involve much larger genes, sometimes it's too large to be included in this small vector. So in the past, or what we're still looking at, is sometimes using a virus that can carry a much larger load, such as a lentivirus, or sometimes they've even attempted in certain trials to break the gene product into more than one and kind of carry it separately using more than one vector. You can also, as I was saying, have non-viral vectors. These are more synthetic vectors that you can sort of tweak and work with to try to best incorporate the product of interest. And then, as you mentioned, you have also antisense oligonucleotide techniques, which are techniques that don't use vectors per se. It doesn't involve a viral vector, but it's working at a different level where it's basically designed to target and complement both coding and non-coding regions of RNA. So you're kind of working at different levels. For vectors, you're talking about having DNA incorporated, and now especially with COVID vaccine, we're hearing about other types of genetic material, mRNA. Transcription RNA. So we know that we can work on a gene therapy from different levels of the genetic coding message.
Just if people are a little bit confused between DNA and RNA, I haven't really figured out a great way to describe it, but you start off with DNA, which is sort of the main message or main instructions. And then in the cell makes sort of basically takes a chapter out of the book potentially. That's like an mRNA molecule. It's just telling you a certain instruction, and that instruction is then made into a product, which is a protein. So what Cynthia is saying is like you can work on the DNA level and change the DNA if there's a mutation, but you can also change that into just the instruction itself. And that's what some of these antisense oligonucleotides are also doing. So sort of getting to what you're talking about your sort of expertise in surgery, can you maybe tell us how some of the gene therapies work? So what exactly, if somebody is getting a gene therapy, what is the process by this happens?
Right. So for now, the approach to deliver the gene therapy product is through surgery. Usually I explain it in the sense that, a lot of people ask, well, can I get this gene therapy in a drop form? Or can I get a pill and, I just take the pill and voila, the gene therapy is accomplished. So usually, I tell them, first of all, you want this product to be targeted. That's really the key word. You want it to work exactly where you want it to work. And for things such as drops or oral administration, obviously the drug has to go through the whole body, or in the sense of topical drops, it has to penetrate all the superficial layers of the eye before it gets to the final area of interest, which is retina. So in order to bypass all these, hurdles, if you will, to really work exactly where you want it to work, the current standard approach is to administer the gene therapy, which is suspended in a liquid form, under the retina. So most of the trials, or including Luxturna as a product, as you will see, is labeled for subretinal injection of the medication. So you're using a very, very fine microinstrument needle, and you basically have a syringe that's loaded with the gene therapy product, and very meticulously through surgical approach, you are able to insert that needle or cannula into the space between the retina, the outer layer of the retina, and the retinal pigment epithelium, and that's where you gently deposit the product.
So are there gene therapies for diseases that aren't retinal diseases? Because you mentioned Luxturna, which is for one specific type of inherited retinal disease. Can you talk a little bit about what other diseases they might be studying gene therapies for right now?
Right. So for sure, Luxturna is making a lot of waves because it's first in ocular gene therapy. It's actually also one of the first diseases where you're directly targeting the site of the diseased gene. So what I mean by that is we also see a lot of gene therapy that's being applied in fields such as hematology and oncology, because those are fields where obviously you do have a lot of, there's the high number of patients, and there are a lot of conditions that are also associated with genetic mutations. What's a little bit different for eye versus these other fields where they use gene therapy is oftentimes in these other fields, you're trying to, you're not necessarily addressing the root source of the disease through the gene therapy, because here the idea is that by delivering the correct genetic copy, you're able to reverse or stabilize the visual loss process, and that's really addressing the root cause. Versus in some of the other fields where gene therapy is used, it's to make a protein or a genetic product that helps with certain aspects in the pathway of the disease. Doesn't necessarily mean that the person who has a certain oncological or hematological condition is cured at the base, but that they're getting supplemental gene products that can help in the modulation of their disease. And I think that stems from the fact that in some of these genetic mutations for retinal diseases, it's a very localized presentation, right? So the disease is only purely a manifestation of the eyes. The problem that's manifested is vision, versus some of the other conditions where you have, for example, problems through circulation throughout the whole body. And, it's harder to target one site in one pathway when it involves different organ systems, et cetera. But here, because vision is the primary target site, we are lucky in the sense that we can have access to gene therapy that targets specifically those specific cells that are damaged. And that's the only manifestation functionally of the problem from the gene mutation.
So yeah, I think what you're saying is that because you're looking at a very small set of cells that have this mutation, the gene therapy for the eye is really trying to fix that one mutation. It's not necessarily, so it's being really, really targeted for very specific mutations in the cell. Is that correct?
That's correct.
Yeah.
Yes.
So how about for other eye diseases that aren't retinal diseases? Are they looking at gene therapies for instance, say corneal disease or for the optic nerve or other things like that, other diseases?
Right. So in terms of other fields of research, there is some work, I think all fields, all subspecialties in ophthalmology have genetic therapy being looked at. In terms of progression, I think for sure retina diseases are amongst the most heterogeneous, have the most different types of mutations that have been discovered and known and have the most advanced progression in terms of research. There is also research in terms of optic nerve diseases, most concentrated on Leber's hereditary optic neuropathy. I know that there are a lot of people who also have who are affected by other conditions of optic nerve hypoplasia or dominant optic nerve atrophy. Those are being looked at, but the research is ongoing, but it's not as it hasn't progressed to such a point where there are specific clinical trials that are ongoing or at the point where recruitment is starting yet. So. Yeah.
Okay, that's really interesting. Actually, it leads really nicely into my next question, which was about how do you bring a clinical trial from sort of a research point of view all the way through to a clinical trial? Like, what is the process?
So basically, the question of from bench to bedside, right? I think it's quite a complex procedure and steps of how it works. I think most people who are on the clinical side, because either they have the disease or they're interested in development, they see sort of once the clinical trial is fully engaged in recruiting patients and on that side, but there's a lot of behind the scene work that happens before that, right? Because it has to go through rigorous clinical studies before it goes through all the phases of human clinical studies, as we know. So usually you have to start, at the very basic level, at the cellular level. If you find a gene, first of all, isolate the gene. Then you look into possibly having the gene therapy that can counter that gene mutation that's found. You need to find usually an animal model first that appropriately represents that mutation to see if you can successfully treat or reverse the effects of that mutation in that model. And depending on what model you start with, usually you have different levels or hierarchies of animal models. You would start usually with a mouse or a rat model that then would move towards other levels of mammals, or be it canine or a dog model, a monkey model, etc. And once you've gone through those steps, then that's when you start going through the human clinical trials. Usually, in the past, these were steps that takes several years, and we're hoping that because this first gene therapy has come all the way from beginning to end over almost 20 years time, that this will accelerate the next steps of clinical trials going into approval, into approved treatment, because the road has been in a way paved, right, for a first run, and we know what are the steps to get to clinical trial. But in essence, if I answer it from a different way, patients who say, I have this condition, how do I get involved in a clinical trial? Well, first of all, we need there to be an initiative to start looking at a clinical trial, because the strength of clinical trials is really in numbers. And because these are, they're rare diseases in the sense that oftentimes there's so many mutations. So each person who has the disease is rare. Obviously, as a whole, we see that many people can be affected by retinal dystrophy. It's just that, it's each gene is rare, but taken together, it's quite a big group of people. But we need to specifically, find clinical trials targeted towards a specific gene And that's, that takes numbers in terms of gathering participants from different institutions, different locations, and that's how you build up a clinical trial from the ground. But someone who goes to their ophthalmologist and says, I have this condition, I want to go through to a clinical trial, we really cannot start necessarily a clinical trial from that site if that's the only patient, or if there are no active trials that are recruiting at that time. We can obviously start the process, but it's going to take time to build from that initial interest to becoming a clinical trial that recruits actively and goes towards treatment phases.
You really put it into perspective about how much work it takes to develop a gene therapy. Like, there has to be so much of this pre-work done first before you can even consider doing a clinical trial. And what you were saying really reminded me, and it's sort of a plug back to what Sherry was talking about why we think the patient registry is so important, is if we have more people signed up for the patient registry, sort of putting their hand up and saying that they have this gene mutation or this type of disease, it makes it potentially easier for researchers and pharmaceutical companies to know that there is need for a new treatment, but also to come to Canada because there are enough patients to be involved in a clinical trial. And also, I know Sherry gets a lot of questions about, I have this gene mutation or this disease, is there a clinical trial? She isn't able to tell you if you can join a clinical trial, but she can help you find out if there are at least any trials that might be of interest there's a pretty complicated website called clinicaltrials.gov. She can help you navigate through that. So please contact the Health Info line. And of course, talk to your eye doctor if you have questions about any specific trial. And so the last question is sort of talking about trials as well, is how do people know if they are eligible? So obviously it's important with inherited retinal diseases or inherited diseases to have their genetic testing, but are there other things that you would look for when you're recruiting for a clinical trial?
You mean if the patient is interested in participating to know that they're a good candidate?
Yeah, so does anybody who has a specific gene mutation, are they eligible, or are there other things that they have to look out for?
Yeah, so for sure, I think signing up to the FCC newsletter is very important, because I think, as much as we said, as we have information, it's very important for the person who knows they have this condition and who have their genetic mutation to also keep an eye out for development, because as we've said, we in Canada, we're at an exciting time where we're trying to build up our network, but obviously we also hear news of different trials that are going on both on a national level but also internationally. We know through the news there are a lot of clinical trials happening in the US, there are many in Europe, in Asia. So I think being aware from the baseline for oneself's condition, that's the first starting point, just to be interested and also to keep updated. I'm sure, the people who are keeping track of all that comes out in the exciting scientific domain and the world, some of them are very well versed in what's going on, and sometimes probably they hear about it firsthand at the same time, or sometimes perhaps even earlier than their own ophthalmologist. So based on that, if someone who has done the research, who knows their mutation and know about their current vision and their visual potential, comes forward and has already sort of done all the background research, then I think that makes the process of choosing them, if they do have a gene that's eligible for clinical trial, easier, because they kind of know the basis of how it works, what gene therapy means, what kind of follow-up or testing of surgical procedure it involves. So that makes the choice much easier. In terms of people who have know they have a genetic mutation in the retina, but they don't know their gene yet, they only have a broad sense of what gene therapy is. I think in those patients, it's important to take the time to really walk through what's part of what we're discussing today, but on a more extended level, on over several visits, so that they have a good grasp and can have time to sort of gather the resources to better understand it on their own time and period. I think in general for now, the criteria for people who are included in clinical trials is quite flexible, in the sense that there are studies involving adults, but there are also studies that can look at the pediatric population. So age is not necessarily a limiting factor. Usually it's important to have no other comorbidities or other ocular problems other than the inherited retinal problems. So it's ideally not someone who hasn't had any other ocular surgery or histories of complex problems that have led to vision loss, because then it would be very hard to kind of delineate what process of the vision loss is from the genetic condition and what is from other causes, for example, trauma or retinal detachment, because really that's also something that I think people in this community at FBC understand, but a lot of people from the wider community come and say, well, I've lost my vision. Can gene therapy cure this? Obviously, gene therapy is not, it's really promising, but it's not an answer to all types of vision loss, right? It really has to be targeted towards gene mutations. So if someone comes with vision loss from a traumatic cause or from a vascular disease, that unfortunately right now is not addressed by gene therapy. So I think that's something that people have to keep in mind when they seek out gene therapy for themselves.
Sorry, I just put myself on mute because the garbage trucks were going by. No, thank you, Cynthia. I think I think it's really important to, we're always trying to offer hope about gene therapy, but it also is, there are some people it won't work for, or some people who there isn't a gene therapy available yet. So sort of important to put that into perspective a bit. So that is it for the questions I had from sort of FBC's perspective, because as I said, we really want to give time for the audience to answer questions. So before I throw it back to Morgan, I'm just going to talk really briefly about a new advocacy initiative that we are going to be launching soon. I just wanted to tell all of you about it today. So as Cynthia mentioned, Luxturna is the first gene therapy for an inherited retinal disease, or for any eye disease actually. And it has been approved by Gene Canada. So it is available to be sold in Canada, and it was approved in 2020. But the next step, there's many, many steps in drug approval processes in Canada. And the next step is that the provinces have to negotiate on a price for Luxturna. So we've been currently waiting for about 6 months for this negotiation to take place. And we know that every day, every week that goes by is another day, another week where somebody might be losing their sight who might be eligible for Luxturna. So we really want to put some pressure and really explain the urgency of this to the decision makers, to the provincial premiers, to the ministers of health, and to the chief negotiators. So what we're going to do is we're going to be launching an email writing campaign in the next coming days, and we will send out some information to you, and we really hope that you will step up in a very easy way, send an email to these decision makers and let them know how important it is that innovative treatments like Luxturna get approved and get funded as quickly as possible, so that nobody is losing their sight waiting for the bureaucratic process to go through. So stay tuned for that. And if you have any questions, feel free to reach out to Morgan, and she can send any questions over to me, and I'm happy to answer that. And I will go throw it to Morgan right now. Thank you.
Thanks, Larissa. That's great. So we do have tons of audience questions, and we're going to get to those in a sec. I'm going to give Dr. Candice a short break. And I would like to introduce all of you right now to my colleague, Jamie Alexanderson, who's going to share a bit of information about how you can get involved in supporting exciting vision research.
Thank you so much, Morgan. Hello, everyone. My name is Jamie Alexanderson. I'm the Senior Manager of Development at Fighting Blindness Canada. I understand there are over 200 people registered for this webinar today. That's just fantastic. Thank you all for joining us. I'd like to take a few minutes today to talk with you about our monthly giving program. But before I begin, I would like to thank our core supporters who are current monthly donors to Fighting Blindness Canada. I know there are many of you who have joined us today to listen into this informative webinar. Your gifts are making a difference by funding the very best research and the work being done by Fighting Blindness Canada. It really is thanks to your donations. We couldn't do what we do without you, so thank you. Monthly donors are such an important part of Fighting Blindness Canada's community. Donations provide reliable and ongoing funding so future projects and long-term investments in vision research are possible. We invite you to join our monthly giving program today. By becoming a monthly donor, you will be making a tremendous impact. Please consider becoming a monthly donor by $1 a day, $20 a month, or any amount that's meaningful to you. Every monthly donation makes a difference. And not that you need incentive, but today, if you join today, you will receive a free pair of designer sunglasses that were graciously donated to Fighting Blindness Canada by Marchand. To join our monthly giving program, please visit our website at fightingblindness.ca or by phoning 1-800-461-3331. Thank you for your time today and enjoy the rest of the webinar.
Thank you, Jamie. That was great. Okay, excellent. So let's get into all of these questions. It's so engaged, which is fantastic. So let's see, let's start. Let's see, I've got all of our emails here. Let's have a look. Okay, so let's start. I mean, there's a lot of questions here too about specific gene therapies, so or specific mutations. So as Larissa mentioned, if we don't get through all of the questions today, any that are outstanding, myself and Sherry will try to get you some information and reach out to you. Okay, but let's start here. So Lisa, and I think you touched a little bit on this, maybe we could just go back to it. So Lisa asked, she said that her son is 2 years old, and in 2019, he was diagnosed with optic nerve hypoplasia in both of his eyes at 2 months old. And later they had genetic testing and discovered that he has a rare syndrome called Galloway-Mowat syndrome. And she said, in hopes of a cure one day for his vision, I'd like to ask the following question. What sort of research has been done in optic nerve hypoplasia in relation to gene therapy? In particular, she's obviously interested in results for young children.
Yeah, great. Yeah, so those are, I think there's a lot of, I guess I can see very keen interest about specific conditions and, syndromes. And, in this case, like we're saying, we touched upon, so Leber's hereditary optic neuropathy is the field of that involves optic nerves that has received the most attention. Once again, because it's mostly it's a disease that we see in ophthalmology and specifically in neuro-ophthalmology. There's actually quite a few neuro-ophthalmologists who work in the field and in the sphere of inherited eye diseases that are also have a keen interest and or do participate in different clinical trials. So I think that combination of optic nerve involvement and gene therapy could be very interesting and very promising for the future. In terms of optic nerve hypoplasia being one manifestation of a much wider syndrome or condition that affects multi-systems, it is in a way, it works a little bit differently because it's only one site of many different organs that manifests the presentation of the condition. So I think ultimately, those would also in the future would involve gene therapy, but one that would target the mutation, which in this case has manifestations at different levels of the body. And as we were discussing, because it has so many different organ systems that are involved, it does become a more multi-organ complex question to address because you're not necessarily delivering just one set of gene mutation of gene therapy to one specific small site. It has to kind of have an effect body-wide in order to have a full effect. And we're not unfortunately, at that level where gene therapy can be administered over a whole body to have multiple effects on multiple organ systems. And that's why, as we were saying, even for oncology and hematology, the gene therapy is targeted towards one gene product rather than treating, the many manifestations of the condition whole body wide.
Yeah, absolutely. It's so complex. And someone else asked over email, just while we're still talking about optic nerves, they asked if there are any recent develops with gene therapy or stem cells to treat damaged optic nerves. So again, not necessarily like a genetic condition, but if there's damage to the optic nerve.
I think it would depend on what the damage stems from. Are we saying is it ultimately is it from papilledema? Is it underlying condition that leads to the optic nerve being a secondary sort of manifestation of the disease? So ultimately. Or even, for example, glaucoma, you have damage to ganglion cells, and that ultimately also leads to damage to the optic nerve. So, the optic nerve is I like to call it sort of the complex cable of the eye, right? So it's oftentimes agglomeration of many of the other cells of the eye not functioning, or it's a final end result of something within the whole system that's been damaged, or there's an imbalance in that. So I think in those cases, it really depends on what the root cause is, and addressing the root cause of the problem that leads to the optic nerve damage is really ultimately the solution if we could have a direct treatment.
Absolutely. So we also have some questions here about COVID-19, always a hot topic these days. For example, there's a few people who have asked about how the pandemic has sort of affected research. So for example, Bob here is asking, he said, clinical trials for gene therapy for RPGR were in phase 2-3 in the US before COVID kind of came along and derailed things. So what has sort of the effect been on this research? And also, Bob's also asking specifically here, do you know of any trials that will be coming to Canada on RPGR?
No, excellent points. And I think, globally, all research has been impacted in one way or another by the pandemic. For sure, in terms of studies, especially for studies where people were, these are rare diseases, people were traveling from really far and distant places to go into the centers that administered the study, especially if it was cross international borders, that really, as you were saying, Bob, puts a hamper on the speed of how quickly the study progresses. In Canada, we do realize that, I think it was mentioned, some areas have clinical trials in other countries that seem to be at a more advanced stage, but we're working the best we can here as the concerted community to try to bring it up to speed here in Canada as well. So in fact, I think for RPGR actually, so MeiraGTx, which is one of the major companies that is working in that sphere, we are planning in Canada to start not only a natural history study for RPGR, but we're looking at different sites in Canada, notably in Toronto and also in Montreal, starting to participate in also a phase 2-3 study for RPGR, hopefully by the end of this year. So we know that there is a gene therapy that is in clinical trials, and there are talks with the company to bring that to Canada, as we do have a large population of patients with RPGR mutations.
That's really exciting. Now, if people are interested in learning more about particular clinical trials such as that, what would be your advice to them?
Depends, on their on their geography. I think obviously reaching out to their eye care health provider, and I'm sure everyone is established locally with their visual rehab centers or with their ophthalmologist, and also of course through FBC, as I've listening into all the conversation that was happening before and in between our discussions. There's a lot of good knowledge about what's going on through FBC as well, that I think people who are registered with FBC or who want to gain additional information can get a lot of from talking to all these resources.
Absolutely. The more resources you have, the more knowledge you can get and find out about all these trials that are coming. Another question that we've been getting to about COVID, and I noticed someone did put it in the Q&A. They're asking, are there any side effects of the COVID vaccine, particularly for RP patients, but we could even broaden that to eye health in general. And what are your thoughts on the vaccine? Would you recommend people get the vaccine?
So there, first of all, for COVID and eye, that has been looked at, especially since the beginning of the pandemic. There doesn't seem to be any direct correlation between eye disease per se and COVID, just to kind of start off, except for the fact that, you know, in the initial cases, we do note that exposure to the COVID droplets could be passed through eye if there's no direct eye protection. But, you know, from the studies of, you know, superficial eye, cornea, and deeper structures, there didn't seem to be any advertent effects on the health of the eye itself. In terms of the vaccine, once again, you know, we've been hearing a lot about different things in the news, mostly about hematological clotting disorders from the different vaccines. I think, you know, obviously, there's no major reports of any major ocular or eye findings or side effects from using the COVID vaccine. That being said, of course, this is still very early on, and you know, we're just getting the first group of people. We're just seeing the first people getting vaccinated, and we don't have much more than, I guess, 4 to 6 months, you know, of data on those first set of people who've been vaccinated. But you know, that being said, I think taking public health measures into account, if we're looking at the global impact of having the vaccine and, you know, the health benefits of avoiding the severe side effects of developing COVID, I think it's reassuring enough to not have to worry about having RP or another underlying eye condition as a major factor to consider before deciding to get the COVID vaccine. So I think, you know, from a general health perspective, it's as Health Canada has recommended, it's recommended to get it. And so far we haven't seen or heard of any major reported eye findings, although obviously we're keeping abreast of the clotting issues as the news roll out.
Sure. Okay, let's see here. So of course, we have lots of questions about specific conditions. So let's just kind of go through a few of them. So Jacqueline's asking, and a few people actually are asking about cone rod dystrophy. Are there any sort of trials or timelines for potential treatments for cone rod dystrophy? And I guess the other question here was, are there any known long-term side effects from getting gene therapies?
Excellent. So, you know, in terms of cone rod dystrophy, you know, again, like, you know, RP, there are many different genes that are involved in the process. And, you know, so it really, as we were saying before, each clinical trial is really kind of what I call a lock and key system. You know, if there's a specific gene mutation in question that's being targeted, then we need to look at that specific gene therapy for that mutation. So cone rod dystrophy is a part of inherited retinal diseases, but it also has different genes that are involved in that process. And, you know, it's, as we know, like even people who work with this all day long, it can get confusing because some of the genes sometimes can produce cone rod dystrophy findings, and then other times it causes, you know, like other macular dystrophy. So it really is dependent on the mutation itself. And cone rod is obviously is a term we know what the disease manifestation is, but we really need to get to the genetic mutation level to know exactly what clinical trials are going on. And there are, I would say there are small studies that are going on, but again, it's, you know, depends on what the mutation in question is to be able to address it on a more specific level.
Yeah.
Oh, and sorry, the other question, right, was long-term effect of gene therapy. So again, you know, the longest data we have is from the trials for Luxturna. And, you know, some of the earliest studies, human studies for those date back to the early 2000s. So we have about, or a little bit over 10 years of data on how those patients have been doing. And so far, you know, from the publications about the long-term effects, it's been tolerated very well with no safety concerns after that period of time. But hard to say, you know, in 50 years or 60 years, is that going to be still the case? You know, we're only working with the first generation of people who've received this treatment. So I think only time will tell, but it's reassuring that so far so good.
That's great. And just changing track a little bit here, we have a couple questions about the age of the patient when they receive the gene therapy. So in the chat, we have a question saying, will treatment be available to older people when the province finishes negotiating with Luxturna? Apparently, this person's specialist had said likely not when they met with them last year. And then Marie is also asking, has gene therapy, is it more successful or effective in younger people than older people?
I love these questions. Thanks for grouping them. I think it makes great themes of, what people are thinking of, kind of the beginning and the end of the the brackets of what's the best time and what are age limits. So I did. I'm in Quebec, so I did participate in the provincial discussions about what age group or what are the criteria for inclusion, and the understanding is, because the disease is very variable, I'm saying not only for RP65, which we know starts by affecting most or almost all patients before the age of 18. It's a pediatric onset presentation, but it's hard to say, who's going to present at what age, at what stage. It's possible to have someone who's very young and have very severe disease, and someone who's older but have relatively less severe presentation. So I think in terms of how the approval will work, and once it is approved, who will be able to get it, I don't see an advanced age or necessarily a certain cutoff necessarily for how bad a vision needs to be before. There are no such criteria from the discussions we've had, for at least different provincial discussions in which, there will be an age cutoff as determining if someone is too elderly to get the treatment because they've had too much damage. That being said, we do know that most likely, yes, the less advanced the disease is, and hence the younger the patient is, there's probably more room and more of a window period to save more vision. For sure, I think from a realistic point of view, if your vision is already very severely affected, you're a candidate for the gene therapy, but the likelihood of how much vision you will regain is, especially if you had the disease for a much longer period of time, is probably less than someone who's a pediatric patient who's just starting to, to have visual symptoms. So but we don't have clear cutoffs of what that ideal window of opportunity, if you will, is. There's no clear data in literature or in different cohorts to say that after vision is at such a certain level, or after we've lost such a certain number of cells, that it's no longer useful or no longer viable to go through with gene therapy. But for sure, if you have more reserve, most likely you will gain back more. But at the same time, if you there's no perfect window, right? Because if the patient is very young and doesn't have symptoms yet, you also don't want to introduce a therapy that, although has kind of proven its safety, there are still complications or side effects that could be related with the surgical administration. There could be a possibility of inflammation. So those are things that can also cause a decrease in vision. And if you're starting off from a point where your vision is asymptomatic or almost perfect, you don't also want to do it too early when you might incur like a loss of cells or vision. So, it's kind of that in-between period, and it's very hard to determine when's the best time.
It's a balancing act, right? Of figuring out
Yeah.
when the right time is. And so we have a question, again, kind of going off of that from Jerry, just to clarify, his sort of overall thing is gene therapy mostly targeting the preservation of existing retinal function, or is there hope that gene therapy could reverse and restore lost function?
I think it's both. At the baseline, we want to preserve, but through different studies, we've seen that there has been regain, especially with the phase 3 Luxturna studies, where we see that people had really a significant increase in their results in terms of the multi-luminance mobility testing. So there is regain, but ultimately, I think it's important to set realistic goals in that we want to preserve so that there's no more loss. But if there is, in addition to that gain, that's kind of an additional bonus, but I think that speaks to that window of reserve, that we're talking about. If we're catching the condition early enough, then it will reverse. But perhaps once we reach a certain level where there's been a certain percentage of cells that are affected and whatnot, it's perhaps more just kind of a leveling off to prevent further, decrease of function.
Definitely. So I think, Larissa, you mentioned this a bit at the beginning, talking about the different sort of types of gene therapy. So we have a couple questions. Leonard specifically asking about a shift from, specific genes to optogenetics. So I was wondering if you could clarify a little bit about what optogenetics means, and if you could explain that type of therapy. And then Bill's also asking about other things like protective factor, gene editing, CRISPR, some of the other types of gene therapy, if you could clarify a bit about what they do.
Right. Some very very well informed members. So that's really nice to see people who are reading up and know so much about the field. So gene therapy is what we've been discussing a lot about, but it's also encouraged to know that there are different approaches. So the ones we've talked about are all gene specific. But then the ones that were mentioned in the chat, some of them, including the optogenetics, the sort of neurotrophic factors, these are treatments that are not necessarily, they don't need to target a specific gene mutation. It means that for people who know they have a inherited retinal disease, these are all factors that can try to enhance or bypass certain of the cells or photoreceptors that are defective or at a diminished level of function. And these are all good alternative therapies which can treat not just one specific type of gene mutation, but different types of mutations. So in a way, we can see them as being complementary, but at the same time, it's less specific because you're just trying to enhance and give factors that try to enhance either the cells that you still have or that bypass the ones that are not so functional, but you're trying to stimulate the remaining cells, for example, optogenetics, where you go through the ganglion cells, but the signal is quite different from what the normal transcriptional steps of what phototransduction from photoreceptor all the way to the optic nerve and to the brain is. So you're trying to use new alternative pathways of stimulating. And I'm saying this coming from the background in the sense that I was also involved in the surgical procedures for the retinal prosthesis. So, I think we often see this graph of where treatment is, where, gene therapy really targets ideally the specific gene mutation at the very, very early stages of the disease. And then you have ultimately the people who are very severely affected that basically only have very little remaining function, but they can be aided by something, such as a retinal prosthesis, where you basically, again, working with the few remaining cells and trying to stimulate those and trying to send off an electronic signal from the retina all the way to the brain. So these non-gene targeted therapies in a way do similar things, but at a level that's much more surgical or kind of more delicate. You're not at the end phase of terminal disease, but you're able to, at the other in the middle of the spectrum, help the visual system regain vision, but through a totally different pathway than the actual cells from phototransduction to the end.
Cynthia, I just actually wanted to say, well, thank you for such a great explanation of really complicated research ideas. I just wanted to tell everybody that we're actually funding research into optogenetics, into some of these more universal gene therapies, so trying to improve cell survival, as well as a gene therapy project about a congenital cornea disease. So if you're interested in any of these projects, please go to our website for more information. I won't take up any more precious time talking about them here, but you can find all the information you need on our website.
Yeah, absolutely. Thanks, Larissa. That's great. Some really great research happening. Okay, and there's so many questions, guys. It's great. It's going to be hard to choose. So we have one question here from the Q&A. So someone's asking, why do we place so much emphasis on gene therapy for photoreceptor degeneration? Could we transplant new rod or cone photoreceptors derived from stem cells into the eye instead? Would that work?
Yeah, great. Yeah, changing a little bit. It's great that we're touching on all these different subjects. So yes, the other, field we haven't really talked about is stem cells. And, so I think, again, the approaches are completely different. Whereas gene mutation, like we said, we're trying to bring the correct copy of the gene into the right location. Stem cells, you're thinking of possibly transplanting healthy cells, viable cells, in replacement of cells that are no longer functional. And that's also something that's evolving in parallel. I think the difficulty with stem cells is that it's, first of all, as you know, the retina is multi-layered. It's very hard to transplant multiple layers of cells into a new location, i.e. the retina, and have multiple layers, integrate well with the surrounding. Most of the research we've been doing with stem cells is more looking at transplanting a retinal pigment epithelium or monolayers of cells. And, the transplanting RP or the retinal pigment epithelium is what's been most developed. But in a sense, the RP is mostly just a scaffold or sort of a support layer for photoreceptors, right? So even though we're successful in that, it doesn't necessarily give us all the intricacies of the functions of the photoreceptor cells above that. And usually what the conditions that have been looked at mostly for stem cells are conditions where you're working mostly with you're working with mostly areas of the retina that have monolayers. So Stargardt disease, for example, has been looked at a lot because the thinking is you're looking mostly at cone cells, or is there a way we can just specifically target one type of cell to transplant rather than having to think about RP that needs to support multiple layers of cells that are degenerating. So, but the difficulty, as I said, is more just how well will these cells survive once they do get transplanted, and also how, if you're transplanting the support layer, how well will it support the photoreceptors with the other layers on top of it. So
you just mentioned Stargardt's, and we have a couple questions in an email about Stargardt's. One particular question we had from Fran was that she said that she'd read that the size of the Stargardt gene poses special problems in the quest for replacement or alteration. Could you talk about that a little bit, is there any progress in the looking at gene therapy for Stargardt?
Right. So yes. So as we were saying, Stargardt is a relatively large gene, just like the Usher gene is a relatively large gene. So for the usual typical adeno-associated virus, it's a little bit on the larger side. People have looked at using, as we're saying, other vectors to work with the ABCA4 gene, which is what was being targeted in Stargardt. So they've looked at a lentivirus. And also, like we said, there are all these new approaches that don't necessarily depend on a classic viral vector, which could be very promising for Stargardt as well in the near future. We didn't necessarily touch upon the intricacies of the antisense or even CRISPR for that matter, but for example, some of the these are being used in different types of mutations, and so the not Stargardt, but the LCA10, the CEP290 mutation, for example, is one of the first studies in ophthalmology and in the all of medicine that's looking at using CRISPR technology to replace mutated genes, and that's an approach that would bypass the classic vectors or the classic problem of packaging larger genes.
Great. So, and we have another question here, a couple questions about, what does this mean, inherited retinal disease, right? So, for example, Geeth is asking, when it said inherited, can a person inherit a retinal problem from their mother's cousin? Like, how do these inherited diseases work in families?
It's hard to explain, right? I guess so. The question is, I guess this person's question is, if no one else in the family has a condition, but just the maternal cousin, can they get it from that cousin? So, to answer that, as we know, genes, we inherit genes from both both sides of our parents, from both mom and dad. And the fact that only that one member of the family has presentation of disease, it's sometimes for disease to manifest, you need one copy of the gene alone to manifest the disease. And sometimes you need copies from both mom's side and dad's side. And then even other times you have these genes that are inherited, the X-linked conditions, which are passed down. Usually classically moms are carriers, and then the male children manifest the disease. So in this specific example, for the maternal cousin to have it, the short answer is it doesn't necessarily mean that that is going to get transmitted to the person in question, but depending on the type of inheritance, it's possible that the mother either has one copy of the gene, or perhaps there are copies that are not manifesting the same way in the mom than the cousin, but it's not a direct maternal cousin transmitting to yourself, but it's if the cousin has it, there's a chance that other members of the family may also carry that gene. It's just that it's not manifesting either the same way, or that perhaps you need more than one copy to have the same effect. But you would have to do genetic testing to identify the gene in question, and also test the different members of the family to see if it runs in the family. So that's what we mean by inherited. That it doesn't jump through different branches of the family tree. If it passes down, it necessarily goes from generation to generation, but it doesn't always manifest in the person that carries the gene. I hope that wasn't too confusing.
No, that was great. So, gosh, let's see here. We're right at the end here. Let's see. We've got lots more questions. And as I said, if we don't get to your question today, we will try to reach out with some answers. But I'd like to finish off. There's a couple questions here about sort of, actually, there's going to be two questions. One, because I think this is an important one. So Janet is asking, is Luxturna still considered a trial?
No. It's considered an approved treatment that should be approved, that is approved by Health Canada, and that should be available to the right candidates free of cost to that person. So I know that's a worry. Sometimes people see it being administered elsewhere and they want to get access to it, but they're worried that they might have to pay out of pocket. And I just want to reassure people who are eligible candidates that you're not alone in this fight for getting access to these medications and gene therapy. We work alongside the whole community to make sure that the government understands the importance of having access to these and of providing these medication free of all costs to the people that it's targeted towards. So, but it's an approved Health Canada product and care.
Again, I'm going a bit on a tangent now because I have more questions, but some other people have asked too, just about gene therapy in general and these innovative treatments. Why are they so expensive?
Yeah, I think because, like we said, people see the clinical side of it, but there's a lot of research, innovation, the technology that goes behind it, that is behind the scenes, that is very expensive to mount a project. And like we said, to have, the sort of patient numbers to generate results, because it's a rare disease, we have to gather data from so many different sources, from so many different places. It's very, I think, labor and time intensive versus something, for example, cardiac diseases or something where you have many hundreds, thousands of patients in just one location. Here, you're kind of pooling all your resources to try to generate the information. And, it's also a long-term disease. So we need time also, the results spread over time to see what the results are over a longer span of time. So I think, all of that mounted together and the technology that goes behind developing these technologies, it makes it, very expensive, both from, a financial point of view and also from a research time and labor point of view.
Absolutely. And I think it's, important to think about too, that like Luxturna was the result of one particular trial, but there were so much research going on in that space that weren't successful, right? So there's so much behind the scenes that goes into making one of these treatments effective and available, for sure. All right, so I'm going to ask one last question. a lot of people are asking again how they can find out more about clinical trials that are happening in Canada. So, you know, we talked to Sherry earlier, get signed up for the patient registry. You can sign up for our newsletter. But Bernie and Jeanette had a very specific question. And they were saying that Bernie has Usher syndrome, and they were really excited to hear that Dr. Kunakoop in Montreal is testing an RNA therapy for Usher syndrome. And so they were just asking, how often will results from this research be shared with the public, or is there any way that people in the public can kind of follow along with the results and find out what's going on there?
So we've also, you know, in view of everything that's been developing really quickly with Luxturna and all these therapies, we've also come together as a group across the country, along through the FBC, and we have a inherited retinal disease committee that's been formed across the country, which includes Dr. Kunakoop, myself, and multiple inherited retinal disease specialists in Canada. So the plan is to actually produce more newsletters and more updates on these findings much more often than was the case before, because the field is advancing so quickly, and we are working on updating, you know, the results of the research on a more regular basis, because we know that now that there are therapies that are available, time is precious, and everyone wants to see, you know, preliminary results or things that can apply to their own condition, hopefully qualify them for treatment. And just also a word, if anyone who's listening in is from the Montreal or Quebec area, you know, and I haven't met yet, if you want to, you know, reach out through FBC, I'd be happy to also, you know, answer any remaining questions after this seminar. Or if you want to meet or have a teleconsultation, I'd be happy to. I'll be available as well. So.
Thank you so much. That's great. And we have lots of questions that we weren't able to get to today, so we will probably take you up on that. Yeah, so I apologize if we didn't get your question, but thank you so much, Dr. Kan, for answering all of those great questions and really giving us a lot to think about in terms of where gene therapy is and where it's going. It's really such an exciting, innovative treatment.